everybody, welcome back to another histology video. Today we'll be talking about question 1 in the embryology series, which is the development or maturation of sperm. So it consists of two phases, spermatogenesis and spermiogenesis, and the process consists of mitotic division of the spermatogonia, and then the meiotic division, and then that turns into the spermatids, and then from the spermatids, it basically moves into the mature sperm, which is motile. And then, as we all know, sperm is produced in the seminiferous tubules containing Sertoli cells, which are these columnar epithelium cells, which are supporting cells because they nourish the developing sperm. And the basement membrane also contains the myoid cells, which basically elicit weak contractions that move along matured sperm. It takes about 64 to 74 days and further 12 to 14 days that are needed for transport through the excretory ducts of the testes and the epididymis. As we all know, sperm production starts during puberty. So in spermatogenesis type A, spermatogonia acts as stem cells, and they divide to create new stem cells and other type A spermatogonia. And after 2 to 3 rounds of division, type A spermatogonia becomes type B, as you can see in the schema, which divide once more to form cells that enter meiosis, turning into primary spermatocytes. And the bridges, as you can see, between the cells remain through the early stages of meiosis, but are lost when the cells mature into sperm. And these connections basically help coordinate their development. And as you can see, the red line clearly differentiates between spermiogenesis and the other part of the development. So basically, spermiogenesis, to put it simply, is spermatids turning into a mature sperm cell, whereas spermatogenesis is basically the whole process, as you can see. So in the schema, you can see the spermatogonia, or the progenitor cells in the spermatogenesis. They are diploid and they contain 46 chromosomes. Through mitosis, they produce more spermatogonia and the primary spermatocytes, which are then going into meiosis. Primary spermatocytes briefly break through the blood-testes barrier, which are tight junctions, moving from the basal towards the luminal compartment. And as they do this, they replicate their DNA and enter meiosis 1, where they pair up the chromosome. The first division, results in 2 haploid secondary spermatocytes, as you can see, and each with 23 chromosomes. And then meiosis 2 happens quickly, which is basically the splitting of the chromatids into 2 smaller haploid cells, or N number of chromosomes. And these spermatids are still within the Sertoli cells near the tubulumen, and then they undergo changes needed to become more mature, more motile sperm capable of fertilization. So in the schema on the right, you can see that there's the acrosome at the top, which contains hydrolytic enzymes needed for fertilization. And then at the bottom, you can see the centriole basically forming the flagella because it produces the microtubules. And then you can see the mitochondria, they form in the middle near the head, used for energy. And after that, you can see that we form the finally mature sperm. So here's just the order of the sperm transport. So we start off from the seminiferous tubules, and then we go into the straight tubules, and then we go to the rete testes, and then after that we go into the efferent ducts, and then the epididymal ducts, and then the vas deferens, and then the ejaculatory ducts. And here is just a schema of how things would go. And yeah, thank you for watching.